Hello everyone, this is Manisha Sansiya from Dr. Hari Singh Gaur Central University Sagar. My topic is urea production. So today I'm very excited. I'm so glad that you could join me today. I hope you all will enjoy the presentation. So in this topic we discuss about first of all of course introduction, then structure of kidney, then step involved in the urine formation, that is glomerular filtration, tubular reabsorption and tubular secretion. So here I am starting with the briefly introduction, that is how actually the urine formation take place. Urinary system function as a filtration of blood and remove waste material from the body. Urinary system, also known as urinary tract, consists of kidneys, ureters, bladders and urethra. The kidney are the blood filtering organs. Kidneys filter unwanted substances from the blood and produce urine to produce unwanted substances. The three-step process clean the blood and create waste product called urine. The urine exits the kidney and move down the ureter to the bladder. As the bladder fills, it interacts with brains and nerve contract and relax the muscular structure and urine is pushed out through the urethra in a process called micturition. In this slide, we talk about the structure of kidney. Kidneys are the major excretory organ in humans. Its primary function is urine formation. Kidney normally can be divided into three different structures: renal cortex, renal medulla and renal pelvis, and finally there is a ureter, that is attaching the kidney to the urinary bladder. Ureter carry urine and urine is stored in the urinary bladder and then released. Blood enters the kidney by renal arteries and after filtration of the blood it leaves from renal veins. Renal artery into the kidney divide into smaller arteries and then arterioles. The arterioles get into the connect with the functional unit of kidney called nephrons. This is where the blood filtration and urine formation take place. The filtered blood then passes through the veins and passes the kidney via the renal veins. The urine is collected in the collecting duct and leave the kidney via the ureters. Each kidney contains over millions of nephrons. And nephrons are the basic functional unit of kidney and help in urine formation. Nephron consists of glomerular capsule or Bowman's capsule and a long renal tubules. Long renal tubules consist of proximal convoluted tubules, Henle's loop, distal convoluted tubules and collecting duct. Urine formation involves three major processes: glomerular filtration or ultrafiltration, tubular reabsorption and tubular secretion. Glomerular filtration. The first step in the urine formation is the filtration of blood that take place in the glomerulus, hence called glomerular filtration. Glomerular filtration is the transport of materials and dissolved substances from the blood to the glomerular capillaries into the Bowman's capsule. A glomerulus is a network of capillaries that resides in the Bowman's capsule and function as a filtration in a kidney. During filtration, blood enters the Bowman's capsule via the afferent arterioles and leaves by efferent arterioles. Not all the component of blood will cross the capillary walls of the glomerulus. Blood cells and plasma protein are too large to pass through the glomerulus capillary wall. Water, salt, hydrogen ions, vitamins, bicarbonate, urea, glucose, amino acid and drugs are across the capillary walls of the Bowman's capsule. The pressure of the blood into the glomerular capillary forces the blood to pass it through three layers, namely endothelium of blood vessel in the glomerulus, the basement membrane of the glomerulus, the epithelial cells of the glomerular capillaries. The endothelial cells has relatively large pores in which solute, plasma protein and fluid can pass through, but not the blood cells. The epithelial cells in the Bowman's capsule, also known as podocytes, intricately arranged leading a few minutes opening called slit ports or filtration slits. These slits on basement membrane help in the filtration of blood and almost all the constituents of plasma except the protein pass into the lumens of the Bowman's capsule. Tubular reabsorption. The second step in the urine formation is tubular reabsorption. The movement of water and solute from nephron tubules back into the circulation is called tubular reabsorption. It occurs from nephric filtrate into the blood in peritubular capillaries. Reabsorption is performed by tubular epithelial cells present in different parts of the renal tubules through active and passive mechanism. While the substances in the filtrate such as glucose, amino acid, vitamins, sodium are absorbed by active transport, while nitrogenous base and water are reabsorbed by passive transport. As we know that 180 liters of glomerular filtrate formed per day and the amount of urine released is just 1 to 1.5 liters per day. This is because about 99% of filtrate is reabsorbed in the renal tubules during the process of reabsorption. Blood enters by afferent arterioles and leaves by efferent arteriole. Efferent arteriole then form the vasa recta, which are basically the capillaries which follow the tubes of nephron. The vasa recta allow the reabsorption of things back into the circulation. After woman capsule, we have PCT, that is proximal convoluted tubules, the Henle's loop, the distal convoluted tubules, and collecting duct. In PCT, the reabsorption of sodium, chloride, potassium, glucose, amino acid, and bicarbonate take place, as well as of course water and urea. In descending limb of loop of Henle, water reabsorption take place. In ascending limb of loop of Henle, sodium, chloride, potassium reabsorption occur. The loop of Henle having descending and ascending limb. The distal convoluted tubules is responsible for the reabsorption of sodium, chloride, potassium, calcium, magnesium, bicarbonates. The collecting duct here is responsible for sodium, chloride, urea and water reabsorption. Now let us focus on the different section of nephrons where reabsorption take place. In PCT, sodium gets reabsorbed together with the glucose and amino acid from the vasa recta via transporter. There is a transporter which exchange a sodium for hydrogen exchange. The sodium reabsorbed inside the cell and exchange potassium by the sodium-potassium ATPase pump. The PCT is important in regulating the acid-base balance. It is a site where hydrogen ion can be secreted. Now what happens? There is a chemical reaction take place. Hydrogen ion inside the lumen of nephron react with bicarbonate ion to form carbonic acid through the enzyme carbonic anhydrase. The carbonic acid is converted into water and carbon dioxide. So CO2 can diffuse back into the circulation. Then same reaction can take place with the same enzyme, that is carbonic anhydrase, which make carbonic acid. And this cycle is continued. This is why there is increase in CO2 level, because there is shift of more hydrogen ion production and hence more acidity. The bicarbonate can reabsorb back into circulation with sodium ion, increase the pH of blood if alkaline. After reabsorption, tubular secretion take place. That is the last step of urine formation. Tubular secretion is the transfer of material from blood capillaries to the renal tubules. It occurs mainly by active transport. Mainly urea, uric acid, creatinine, and some drug, hydrogen ion, ammonia are secreted in proximal convoluted tubules. In the loop of Henle, urea is secreted. In the distal convoluted tubules and collecting duct, hydrogen ion, potassium ion, and ammonia is secreted. After all the reabsorption and secretion, urine is produced. So what is in urine? Well, it's mainly a water, also contain nitrogenous waste. So here is the reference of my presentation, Human Physiology by C.C. Chatterjee and S.C. Rastogi, biorender.com, byjuice.com, slideshare.com, YouTube Armando Hossain Video, googleimage.com. I hope you all enjoyed and liked my presentation. Thank you.